there's a mystery at the heart of life on Earth. At the molecular level, biology is asymmetrical. Most organic molecules have a mirror image twin. But biology doesn't use both twins. It tends to pick one mirror image over the other. Like left and right hands, mirror image molecules can't be superimposed.
You have to use a glove for your left hand and a different glove for your right hand, otherwise they will not fit. And that spatial property turns out to be very important in biology.
This property is known as chirality, from the Greek for handedness. Chiral molecule pairs are made up of the same atoms arranged differently in space. Depending on their molecular geometry, they are classified as right-handed or left-handed. Chemistry produces both right-handed and left-handed molecules. Biology's preference for one over the other is called homochirality.
Life is homochiral, which means all biological molecules of a certain type have one and the same type of chirality in all of life.
Chemistry seems to respect symmetry, so why does life reject it?
Why did life settle on the chirality that we see right now on Earth? Is this the only chirality possible for a living system?
This is one of the big remaining mysteries, despite the big progress we made in 20th century in molecular biology.
Biology's broken mirror has taunted scientists for centuries. Research into chirality could reveal how life began, whether it exists in other worlds, and some scientists fear pose an unprecedented risk to its existence on Earth. In 1848, when famed chemist Louis Pasteur was a graduate student in Paris, he made the first of his world-changing discoveries. Pasteur was studying the chemistry of tartaric acid, a crystalline byproduct of wine production that's also involved in many biological processes. When he synthesized tartrate crystals in his lab, Pasteur noticed that half of the crystals had edges that pointed left and half pointed right.
He could see actually that they looked different by eye, and the relationship was mirror image.
Pasteur exposed the crystals to polarized light. He found that the right-handed crystals rotated the light clockwise, while the left-handed ones rotated it counterclockwise.
And he made the connection that whatever asymmetrical properties embedded in these crystals made them behave differently when they interact with polarized light.
Based on this insight, Pasteur believed that the mirror image crystals were made of the same molecules, but that these molecules were arranged differently in space.
And that's where the left-handed, right-handed chirality came from originally.
He had discovered molecular chirality. And he noticed something else. While the tartrate he produced in his lab yielded a 50-50 mix of right and left-handed crystals, the biologically produced crystals were exclusively right-handed.
And that was, in fact, the new big discovery. Life isn't like the inanimate world. Life was special in the way it was homochiral.
Only products originating under the influence of life are asymmetrical, Pasteur wrote. At the time, Pasteur's discovery seemed like an obscure curiosity, but nearly a century later, a tragedy rocked the world.
When we started using synthetic organic chemistry to synthesize drugs, many organic chemistry processes result in the mixture of both chiralities of a given molecule. And often only one of those chiralities has the correct biological activity.
In the 1950s, scientists marketed a drug called Thalidomide to pregnant women. The mixture contained both forms of the molecule. One was known to relieve nausea, and its mirror image form was believed to be inactive.
People thought that's okay, because if the opposite chirality has no biological function, then it won't hurt to take it. Well, it turns out it does hurt it.
The mirror image molecule caused congenital anomalies in developing embryos. As a result, more than 10,000 children were born with deformities, such as malformed arms and legs. The thalidomide disaster forever changed how we make medicine.
People start to realize how important chirality is for the biological functions.
The thalidomide disaster renewed interest in the molecular basis of Pasteur's discovery, resulting in a field called stereochemistry, the study of the spatial arrangement of atoms that form the structure of molecules. Most biological molecules have something called a stereocenter. Usually this is a carbon atom. Many molecules have more than one stereocenter. The stereocenter is surrounded by 4 atoms or groups of atoms. These components can attach to it in mirror image configurations called enantiomers. If enantiomers don't share a plane of symmetry, meaning they can't be rotated to match in space, then they are chiral. Chemists have a set of complex rules to determine whether a chiral molecule is deemed right or left-handed. This molecule is right-handed, denoted with an R for the Latin rectus, meaning right, because the atoms attached to its stereocenter orient clockwise. The mirror image orients counterclockwise, so it's left-handed, denoted with an S for sinister, or left.
It's not the molecule itself that's left-handed, it's that every chiral center inside that molecule is of the same chirality. When we say a peptide is left-handed, what we really mean is that every chiral center atom in every amino acid in that molecule is left-handed.
A molecule's handedness is core to its function. Biomolecules react through a lock and key mechanism. They fit together. Chirality makes this form of molecular recognition and activity possible. Left-handed molecules unlock certain enzymes by binding with their structure. The right-handed version of the same molecule won't catalyze the same chemical reaction. The process of cell signaling also depends on chirality. A signal molecule has to fit into a cell receptor in order to trigger a response.
One of the receptors that very strongly relies on chirality of small molecules is olfactory receptors, so the smell receptors. And there is many molecules that smell different depending on which enantiomer of that molecule you use. A great example is a molecule that's very commonly used in cleaning products. It's called limonen. One enantiomer of limonen can smell like pine needles, and the other enantiomer smells like citrus. It's the exact same molecule chemically. The only difference is stereochemistry.
Left-handedness versus right-handedness really matters. In fact, life cannot really function if it's not homochiral.
Our picture of prebiotic Earth is based on evidence and on assumptions.
Billions of years ago, before there was life, chiral molecules ruled Earth in equal quantities. Then somehow, life emerged, and it was homochiral. Living cells are almost exclusively made up of left-handed amino acids, but the sugars used in biological processes are right-handed. DNA is also chiral. Its double helix winds only to the right.
And it's one of the biggest mysteries of the origin of life. Why?
It's possible that the emergence of asymmetry was a key shift that enabled molecules to link up.
There are lots of experiments that show that if you have a mixture of left-handed and right-handed nucleic acids, the building blocks of RNA, you cannot actually build a functional RNA. And if you don't have a functional genome, you cannot conceive of life.
Nothing would be able to really interact with each other because you would have this mixed system where the lock doesn't fit the key, basically.
The mechanisms that biased the symmetry and resulted in life's broken mirror are the subject of much debate.
The scientific consensus on the origin of homochirality is that there's no consensus.
One theory suggests that a slight excess of right-handed nucleotides might have become trapped in a shallow pool where the difference was amplified on prebiotic Earth. Other theories center around the role of magnetism, an idea that dates back to Pasteur's experiments with polarized light. At Harvard University, Dimitar Sazalov and Furkan Öztürk have demonstrated that interactions between electrons and the magnetic surface of early Earth could have given life its asymmetry. Their experiments utilize a newly discovered effect called chiral-induced spin selectivity, or CISS, which suggests that the magnetic orientation of electrons has an effect on chirality. To test whether CISS could have biased early chemistry on Earth, Sazalov and Öztürk started with a 50-50 mixture of left-handed and right-handed RNA building blocks. They placed this mixture on an iron-rich magnetic surface to simulate the conditions of early Earth.
Dependent on the magnetization direction, either the right-handed crystals appeared faster or the left-handed crystals appeared faster. So we were able to break the mirror image symmetry, and we attained an asymmetric system.
We could see a clear way how this could happen under natural conditions on the early Earth or other planets.
Although the CISS effect is a promising lead, it's one among several.
Until we invent a time machine and actually go back and witness it happening, we won't have a convincing evidence of this particular mechanism was the one that guaranteed homochirality during the terrestrial origin of life.
Homochirality is an essential feature of life, but many scientists believe that life could exist using the opposite enantiomers to those found on Earth, as long as homochirality is preserved. This idea is known in synthetic biology as mirror life.
So for example, if you want to make a mirror bacteria, you would make it with the opposite chirality DNA and the opposite chirality peptides. They would interact with each other just like normal chirality peptides and DNA interact with each other.
Mirror life is still hypothetical, but researchers believe it could be synthesized in just a few decades.
I used to think that mirror life would be a really fantastic research goal. It has biomedical applications. It's possible to imagine drugs that would be stealth to the immune system made out of mirror cells.
But creating mirror life could have severe consequences.
Everything has a predator that hunts it, a virus that can make it sick. Everything is regulated. And if you create a mirror organism, it, in theory, would exist outside of all of those regulatory mechanisms and just grow uncontrollably. And so the only safe way to contain a mirror cell is to not have it.
In 2024, Adamala and 37 other scientists published a technical report highlighting concerns about mirror life.
There's still a lot of fascinating research directions that we can pursue. That field as a whole can move forward without creating a mirror cell.
Nearly 200 years after Pasteur's discovery of biology's broken mirror, scientists are still trying to understand homochirality.
It seems to be so simple, right? It's a geometric feature, and it sounds like we should have an answer by now.
We want to understand what the phenomenon of life is actually.
Uncovering the mechanisms that biased life's asymmetry could reveal not only how life began, but also whether it exists elsewhere in the universe.
We know that chirality is a thing in the universe. When we analyze meteorites, anything that could be chiral is chiral. So if we figured out what is the actual mechanism of the origin of homochirality, then we'd say, okay, according to what we know about the origin of chirality, we think life on Europa, if one existed, should be of this chirality. That will have a huge practical application in designing the instrumentation for the missions. And when we actually go look for life elsewhere, we will know what we're looking for.
This would have delighted Pasteur, who saw a connection between life's asymmetry and the universe. Chirality is one of the links between life on Earth and the cosmos, he wrote.